So welcome back, students. Okay, so anything that you would like to ask from our till now discussion? Anyone would like to ask? Okay, so if there is no query, then we can move further. Okay, so we were talking about different things that are going to affect the migration of DNA. And as we mentioned that DNA have to migrate according to the electric field and the size. And one more term that I want to tell you that all of you might be aware about that. That term is electrophoretic mobility, and that electrophoretic mobility is represented by mu. And all these terms, all of you have studied in your previous semester. So please recall all the things, and these all may become the part of your exam also, because these all you have studied, and all of these things are related to your experiment. Okay, so the next thing that we were referring is the conformation of your DNA. So when we are referring the conformation of DNA, so conformation things that you have studied in case of when we're talking our enzymes also in the supercoiling. So in the supercoiled DNA that we mentioned that when the DNA is going to be circular or it is going to be linear and it is going to be supercoiled or mixed. So what type of your DNA is, what type of conformation it is going to have. So on that basis, your DNA is going to migrate with different speed. So one factor that you can say that it's the size that is going to affect the migration. So, but other than size, if you say that size-wise all are equal. So size means base pair that you are referring. If you are referring 100 base pair, if you are having a 100 base pair DNA, and that DNA may be linear one, stretch linear is there, and second it may be a circular one also. And in case of circular, we also you okay, just to check me whether it is there or not. Sorry, it's in the front one. It might be there with when you're talking about in your theory part. Okay, so in case of the when it is circular, then it may further have different type of conformation. So one either nicked. So nick means one strand is broken, and both strands are broken, and it is going to be linear. So circular one is there, then it is going to be and the circular one is there, then it is going to be super coiled also, and one more type of there it may be a single stranded also. So now these different type of conformations are there. So different type of conformation, they are going to migrate under the electric field under your gel electrophoresis differently. So why they are going to migrate differently? The first thing is the nicked one. When we say term nicked, so what do you mean by the nick? Nicked and circular. So whenever nick is mentioned, it simply means that you should understand it is a circular one. So you should correct it. So here is the nicked and circular. How it's going to migrate nicked and circular? As you can see in your word file, so here the migration is up to down. So nicked circular is going to migrate slowest. So why, can anybody tell me the why it is going to migrate slowest? Can anybody comment why the nicked one is going to migrate slowest? So just I'm moving it to a PowerPoint one, so there we can see it in a better way, we can understand in a better way. Just a moment. So this is the one that we are referring. And as I mentioned, this is gel, and it is going to be, so when you run your DNA, it appears like a band. So band is like this white color thing is there. This image is white, but when you are going to visualize under electrophoresis, under your electric field, you are going to see it orange in color. But when you take it to image, so for image, UV may interfere with your imaging. So for that, we are using filters. So those filters filter it. So in that case, you just see the light, you don't see the colors. So the image that you're going to get with your gel documentation device or your imager, it is going to be a black and white image. Okay, so as I mentioned, the migration is like this. So here it is going to be negative, it's going to be positive. DNA migrate from the negative to the positive because it is going to be negatively charged. This is a nicked circular. Okay, so nicked circular is going to be like this and like this. This is nicked and circular. And linear is going to be like this. Supercoiled is going to be like this. Circular single strand is going to be like this. Okay. These are the order in which DNA is going to migrate. So it's to comment why such migration is. You can take time. So take 5 minutes, think over it, and we'll use it again. Why such type of migration is there when your DNA is of this type? Okay, so I will join back after 5 minutes. So this is a task. So I'm giving you 10 minutes. So don't say 5 minutes. So I'm giving you a task. This is a laboratory task. Simply you can say. So you have to answer after 10 minutes. How you can also give a vague ideas. This may be the reason, and that may be the reason why it is like it is migrating like that. Okay. So after 10 minutes, I will be. You should all must have some vague idea I may say that. Okay.
Excuse me, sir. Excuse me, sir. That you are audible.
Okay, students, welcome back. So hope you analyze this figure and will be able to comment why the migration is like that. Are you here?
Yes, yes, sir.
Yes. So who will comment first? Who will comment first? तो comment करने के लिए सब गायब हो गए. So I'm calling by name. So I hope Manashwini can do something. Manashwini,
Yes, sir.
can you comment?
Sir, I think it moves slowly because when it's been nicked in only one strand, then the DNA it can partially unwind, right? So I feel like that might have affected its migration, but I'm not sure.
Then and how did now this is fully unwind in case of linear. Linear is moving fast, wherever your nicked circular is moving slow, and supercoiled is even moving faster. So here one thing is common, size is common. Size wise, all are alike. So size is not any issue in this case. So if you say it is 1 kb, so if you deal is all are of 1 kb, but all 1 kb they are migrating differently. A little bit you whatever Manaswini you said bit right also. We are going to utilize it. That's what you mentioned. So next I'm going to call yes.
Sir, I would like to add that if the
Yes.
size, as you
Yes.
mentioned, is the same, but the conformation is different in all four of them, right?
Yes.
So the super coil is moving at faster pace because it has additional additional winding. That's why it is called super coil, right? So
Yes, you're right.
so it
Yes.
has the conformation is constricted in this, and the one which is moving at the fastest pace, the circular and single stranded. Even though it is single stranded, it is mentioned that it is circular, so the conformation still remains a little bit confined in it. That's why it moves ahead, other than Nick and the linear one.
Uh, but why linear is moving slower?
Sir, as Manaswini mentioned that the next one has the unwinded part of the DNA, so it is loosened a bit, so that what makes it conformation different from the supercoiled and the circular. That's what
Okay.
I think.
Yes. Whatever you all are mentioning, you are partially right. What you're saying is not 100% wrong. And as Yukta mentioned, the conformation. So what conformation is going to do? As we mentioned that it is a pore size which is going to provide the migration. So through the pore they have to migrate. So as the both the all of are of similar size. So but in the first we are talking about the circular single stranded. So when it is a single stranded one, so then it is bit more fragile. So when you are saying more fragile means it can bend its shape according as it want, because there is no restriction is there. So there is no, as when you're talking about a steric hindrance is not at all there in this case. So because of the absence of steric hindrance, it can take whatever smallest shape it want. Okay, so it can move like make like this. So when it forms such type of structure, what is going to occur? The final size that we are seeing, you are seeing a size was this much, but now size is the space it is occupying is very less. So it can migrate very easily through the pores. So it is migrating easily through the pores, that's why it is moving fastest. Now next coming to the supercoiled one. So supercoiled one is as Yukta was saying, it is supercoiled. So it's occupying a less space. So when it is its hands are not extended one. So it is moving like this. You can say, occupy like when I took an example, when you can squeeze yourself, when you can squeeze yourself, then in that case you can enter inside even a small pore also, like simple that you know about the cat. So cat बड़े साइज की होती है, लेकिन जब भी खिड़की वड़की से उसको निकलना आता है देन वो स्क्वीज करती है अपने आप को, और एक छोटे से एरिया से भी, छोटे से पोर से भी, छोटे से साइज के आपकी खिड़की से भी वो दैट कैन पास. So that is the thing in case of supercoiled one. So now when you are supercoiling it, now your final area, its a real size is going to be become less. Now you can say that, sir, this property can be with the everyone. Yeah, this double standard one can also do, as we mentioned in case of your single one. So in case of single standard one, as I mentioned, steric hindrance is not there. Yes, it can do. It can change its shape like this. So it is changing its shape like this, its size becomes smaller. But its size, as it is a double standard one, steric hindrance is there. It can't change its shape as a single standard one is doing, or as in case of the supercoiling one is there. Okay, so that's why linear one is moving slower than the supercoiled. Now coming to the nicked linear one. So now thing is that it is a double standard one, it is nicked, so it's relaxed, so no supercoiling is there. Now it is a circular one. So because of its circular nature and because of the strict hindrance that I mentioned, because of the double standard one, it can't change its shape. It is going to remain within this frame, this area. Wherever you can say its size is going to be becoming lesser. So that's the reason your nicked circular is moving slowest, because it is circular one. It can't change its shape because of its shape, its topological change in it are restricted. Wherever in case of linear it can do, but it can't do much. Supercoiled one, it's already made a squeezed one in an area that has become very less, so that's why it's moving faster than the linear one. Wherever single stranded circular one, it is, as mentioned, it is a linear single stranded, so then it can move fastest. Okay, so the most important one are these three: nicked, linear, and supercoiled. So now here also in this supercoiled one, as we mentioned that plasmid can be, your circular DNA can be differently supercoiled, and that depend upon the how much linking number is there. It may be -1, -2, -3, -4, so on. So simply you can say the -1 means it is going to be like this. -2, it is going to be like this. Then so on, 3 further increase like this, and 4 like that. Okay, so in these example, same DNA size are going to have different supercoiling. So whichever is the more supercoiled, that is going to be move fastest, which is less supercoiled, that is going to move slower. So within supercoiling, depending upon the level of supercoiling, the migration is of that speed. Okay, so as we know that when we are having DNA or plasmid DNA in your cells, in which it is naturally occurring, as you mentioned, that as you all know that many bacteria, they are having naturally occurring plasmids. So for that Staphylococcus pneumoniae, in which you rough and the smooth strain that you have studied. So in that case, the level of supercoiling in your plasmid, which are naturally occurring, that is going to be different. So every plasmid molecule is going to be differently supercoiled. So if you want to study the supercoiling pattern of your plasmid, which is naturally occurring in your cell, so in that case you will found that there is not just single band, as we are mentioning here, just supercoiled. Supercoiled means one conformation. In that case, you are not having just one band, you are going to have different bands. Even 10 bands, 20 bands might be possible, because your plasmid is going to be larger in size, so level of supercoiling may be different. So you can say that 10 different types of supercoiling might be possible in your naturally occurring plasmid. But when we are talking about our vector that we are having the plasmid, its size is a bit smaller. So in that case, generally we are having these three type of conformation in your plasmid. Either it is going to be nicked or it is going to be supercoiled. If you have treated with the restriction enzyme, then it is going to become linear. So most of the time you are going to have two conformation in your plasmid that you are going to get from your plasmid accession from your transformed cell, and in some case you may have three also, but most of the time you are going to see only two conformation, that is going to be nicked one and supercoiled. Okay, so this is the reason why different types of migration is there, why different in your supercoiling. Okay, so now discussion is open. So if you would like to ask anything from today's discussion, we will continue our lab in our next laboratory session also, in which we will describe how to perform, how the really gel electrophoresis is going to work, and when you are going to be in the campus, then how to perform these experiments. And also in VLab portals, we will see how you are going to make the gel, how you are going to run the DNA sample inside, what different components are going to be there in your gel electrophoresis experiment. Okay, so any query? Any questions or any query through this plasmid gel electrophoresis that we have discussed yet?
No, sir. No questions.
Okay,
No,
so
sir.
there is no question. Okay, so if there is no question, then it is the end of the session. So please turn on your camera while I call your name. Okay, and so in previous class, two were absent. So absentee's name I'm mentioning, one is the Janvi was absent in the previous class, and Tridiv was absent in previous class. So now I'm starting again. So one is Adish. Adish. Not there. Atoiba.
Present, sir.
Present, sir.
Atoiba. Ayushi.
So
Atoiba,
Aditya.
you're not. Atoiba, you're not turning on your camera. Yes, now you're visible. Yes, Demetrius.
Present, sir.
Devanshi.
Present, sir.
Ishita.
Present, sir.
Janvi. Janvi. Final call for Janvi, like in the airport. Janvi. Janvi is absent in this also. Okay, Kshitij.
Present,
Kshitij. Manaswini.
Present, sir.
Namrita.
Present, sir.
Nandini.
Present, sir.
Nehal.
Present, sir.
Nandini, your camera is still off. Rupal.
Present,
Nehal
sir.
Rupal. Sahiba.
Present, sir. Nehal present.
Present, sir.
Shakti.
Present, sir.
Sanjana.
Present, sir.
Shivani. 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 Tanvi.
Present, sir.
And Tridev.
Present, sir.
So Tridev, why you was absent in previous class? So where you were?
So I
नींद आ
was
गई थी
present.
क्या? नींद आ गई थी? नींद आ गई थी?
नहीं सर, वो मैंने किया था बट इट वाज नॉट वर्किंग फॉर सम रीजन.
आवाज भी नहीं आई भाई, ना चैट बॉक्स में चैट टाइप किया तुमने?
हाँ सर, चैट बॉक्स पे नहीं लिखा था बट
There was
मैंने
no
बोला था.
response. There was no
नहीं सर,
response.
था
विवेक. विवेक.
Present, sir. Present, sir.
अब तो मिठाई लाएगा भाई, अब तो आवाज सही आ रही मेरी.
सर मिठाई पक्का लाऊंगा 15 को. कौन सा खाओगे?
युक्ता.
Sir, present.
Okay, so युवराज. Yes, कौन बोला?
Sir, sir,
Present,
can
sir.
you hear me?
Yes, Shivani.
Sir, sir, I'm present. Actually, I wasn't able to unmute myself.
Okay, so in this session, Janvi is absent. So again, I'm calling Janvi. So Janvi is absent. Rest all are present. So thank you all. Stay safe, stay happy and healthy. We will meet again in our next session, and we will continue with our agrogel electrophoresis. And even now, we have still few more minutes. So if any one of you would like to ask quick questions, you can ask. So no questions. So it means everything is a bit clear to you. So thank you all. Stay safe. Stay happy. Good day.
Thank you, sir.
Thank you, sir.
Thank you, sir.
Thank you, sir.